Don't know me, I'm Bird. Say, got it. That's Jessica. We're lucky to have Michael Beauchamp here to speak with us today. And I suppose I should say we're also lucky to have Michael Beauchamp here at the University of Pennsylvania, because he didn't arrive that long ago, in 2021. He came here from Baylor, and he came to Baylor from the University of Houston. And he's got a really remarkably varied research program that studies all manner of different things. But the topic that's most relevant for today's presentation is his work on the neural basis of multisensory cue combination. Many of you know that multiple different senses can provide and gather information about a given environmental property. I can, look at and touch this podium and gather information via the visual system and via the touch system at how thick the top of the podium actually is. You can gather information about location via the visual system and the auditory system. So if you close your eyes, and I'll walk over here, right? And you can determine where I am. If you point, you'll probably get it reasonably accurately. So you can gather information about, nice job, Russell, vision and touch. About the same location and space. He'll be talking about, among other things today, the neural basis of the McGurk effect, which some of you may know by name, but I won't describe it. I'm sure he will. So he brings a lot of different tools from neuroscience to studying these questions, from fMRI to implanted EEG in human patients, if I'm not mistaken. And I'm looking forward to hearing his presentation today. All right, let's welcome him.
Thanks. Well, it's great to be here. I moved here in the middle of the pandemic, so I'm still getting to know everybody, and it's a great chance to. I think my only visit to Penn was right when the university shut down, so I actually never gave a job talk or anything. So you can call us that maybe. Although it's too late not to hire me if you don't like it. So I'll start by thanking the organizers, and I think next month in this seminar series we have Peter Rolsma coming from the Netherlands Institute of Neuroscience, and that'll be a fantastic talk. He's doing a lot of amazing work, so I encourage you to come to January's seminar series as well. And I want to thank all of the trainees and collaborators who helped with all this work through the years, and the current lab members shown down below. And the overview of the talk today is as sort of first definitions. What do I mean when we say multisensory communication? Then I'll talk about some fMRI experiments that we've done over the years, and then some behavioral assays of multisensory integration in speech, especially in neural substrates measured with intracranial EEG in epilepsy patients, like Johannes mentioned. Then I'll talk about some outstanding questions for future research. And I will say this is a little different. Most talks I give are very focused on the most recent stuff from the lab, but I thought today I would give sort of more of a long-term overview, just because that seemed like something different to try. So it's more weighted in some ways to work that it's not heavily weighted, recency weighted. It's more evenly biased across the years. So the traditional approach to understanding communication has been basically to find it as language. So communication equals language, and then you reduce language to its component parts in the traditional reductionist scheme. So you can have a phoneme, which is sort of the elemental token of speech, which say would be raw, and then you could have morphemes, which are words and other larger parts of speech, like the word ranunculus. Then you can have a phrase like, do you have any ranunculus? That phrase only makes sense if you know the meaning of ranunculus. It's a beautiful flower. And then you have the goal of that phrase or that sentence, which is to essentially say, hey, I want to exchange some money for a flower. So that's the meaning of that phrase. Do you have any ranunculus? So this is what's motivated, or thousands of studies over the years, and still probably the dominant paradigm in studying speech and language. Sometimes you'll have an auditory recording instead of just written text, but by and large, it's largely reduced to language. Of course, if we go out in the wild and look how humans actually communicate, it's really nothing like this. So this is a short video from a popular internet fellow named Zach King. So we'll see if we can play this. Some of you may have seen this clip.
Hi, do you have any ridiculous? Oh, I don't know. Let me go check the back for you. You're in luck. You got our last one. Thank you so much. Yeah, sure.
Let's try one more time. Just not just for the sake of this video, but I'd like to see if we can get it to play through the speakers because I have some other auditory clips here. I know this may mess up the people on Zoom, but let's try that one more time. See if it's. There we go.
Hi.
Anyway.
Ridiculous. Oh, I don't know. Let me go check the back for you.
So the point, we won't need to go through the whole clip again, but you can see that the idea here is it's not just, words written or recorded words. There's a lot of other stuff going on. And if you're a visual neuroscientist like me, you're particularly struck by how much visual stuff there's going on in human communications. And we can sort of make a list. First of all, if you just have this text transcript, you can kind of meander through it at your own pace and think about it. But when you see real world human interactions, they're very dynamic. There's a lot going on. It's not sort of one word at a time. Maybe you talk that way to Siri, but when you're talking to other humans, it's always a back and forth. There's a lot going on. Lots of information coming in across many different modalities. Of course, in the auditory modality, you have the speech itself, but you also have prosody, the intonation, the stress, the expression, emotions. And then in the visual modality, you have lots of stuff. Even before I start playing the movie, you can pretty much tell that Zach is here in a flower shop just by the context. You've got spatial location as he moves through the scene. There's whole body movements, there's arm movements, gestures, we call them, facial movements, both emotional and speech. So there's just a ton going on. And it's really interesting to think about how the brain can take all of this incoming information, which is coming at you so rapidly, and integrate it and make sense of it. And of course, that's why multisensory integration is so valuable, because it helps you do things more accurately and faster than you could with one modality alone. So by using this information for multiple modalities together, you can judge what's going on in the scene, what the communicative intent is much faster and more accurately than you could with a single modality. And this begins very early in life with what's called infant-directed speech. When mothers and fathers talk to their babies, they naturally adopt a high sing-song tone. They slow down their speech. They exaggerate their facial mouth movements. So this is something that we learn and something that we continue to learn till quite late, maybe even to adolescence. I'm not going to talk about touch today. I have done some work in touch because I think that's a fascinating modality. It doesn't carry as much information, but it's very important, obviously, especially for affiliative interactions. So how does multi-sensory integration play out? When we have emotional content, we can integrate the facial expression with the vocal intonation. So if we can use sarcasm, say, or we can roll our eyes if we want to visually convey sarcasm. So if I have a sarcastic tone and you see me roll my eyes, you're pretty sure I'm being sarcastic when I suggest that we go to a steakhouse for dinner when we're all vegetarians. But if you didn't have that cue, you might think I was being serious. So we have spatial reference as well. So if I look over there and say, hey, why doesn't someone grab some candy from the table? Everyone's going to naturally orient to the candy, where my eye gaze is directed to the table over there, as well as the verbal cues that I'm giving you. And most importantly for this talk is speech content. So understanding speech is quite a challenging computational task. Even the best, top of the line computer speech models struggle to keep up with continuous human speech because it's coming at you so quickly, 150 words per minute. Each word is selected from thousands, tens of thousands of alternatives. So it's a real computational challenge to make sense of speech, and the visual modality can give you extra information to do that. So again, for the purpose of this talk, I'm going to focus on a turning point, I think, in the neuroscience of human communication, which was the development of bold fMRI. Some of us here remember that development, where very quickly we went from the age of PET. This is an image from Mark Rakel's book and Mike Posner's book in 1994, where you can see it's blurry. The spatial resolution is pretty crummy, centimeters. In fact, sometimes once a PET paper was published that mistook people clenching their jaw for activity in the temporal lobe, because the spatial resolution of PET was that poor that you thought the blood flow to the temporal lobe was blood flow to the jaw was really blood flow to the temporal lobe. Whereas in the very next year, in 1995, Marty Serino and colleagues published a seminal paper in Science showing that you could map out the retinotopic areas very precisely in the living human brain. And this required hours and hours of data collection, which you could never do with PET, because if you're injecting people with radioactivity, you have very limited amount of data you can collect. So that was really a milestone. Of course, we're all familiar with the basics of fMRI. Someone lays in the scanner. In this case, they would actually be in the bore of the scanner, but I have Johannes here laying outside, so you can see the screen behind the scanner, and then the patient or subject sees the screen through a mirror and can wear headphones and can wear somatosensory devices, all sorts of things that you can have people do in the scanner. So what we can do then is measure the bold MR signal, which, as you can see here, if we have a stimulus delivered right in this yellow, in this white region here, the time series in an individual brain voxel increases and then decreases back to baseline over the course of a few seconds as we collect T2 star-weighted images. Now, what that lets us do is measure activity across the brain as people do different things. And when I started graduate school, the dominant paradigm was really, again, a very reductionist paradigm, where instead of presenting real images like pictures of flowers, you would try to say, okay, what's interesting about pictures of flowers? Well, it's their colors. That's what makes flowers so interesting. So let's design a visual task that captures that visual property of color, but is very well controlled. So in this case, we adopted a clinical test known as the Farnsworth-Munsell 100 hue test. And as you can see, there's 5 colored wedges here, and the patient has to decide, the subject has to decide, are they in order by color? In this case, they're out of order by color, which you can only do using the hue information, because we painstakingly match the luminance of each of these wedges so that they're exactly the same. And then you can have a control task where you vary the luminance of the wedges, but there's no color. So everything's the same, except there's color here and not here. When you do that, you see there's these areas on the ventral surface of the brain. So here's a lateral view of the brain. We inflate the brain and flip it over. You can see on the ventral surface, there's a bunch of areas that are sensitive to color and color decision-making. So that's sort of the traditional approach, and then you can apply that to other things like motion or form. So here's a study where, again, instead of presenting an actual moving figure, moving human, which is what we see every day, I spent years making these complicated figures out of random dot kinematograms, where the only cue to the fact that there's a form is that the dots inside a shape are moving one way and the dots outside are moving another way. So these are very important and well-controlled stimuli, but they don't really, they're a very long way from what we see as we're walking around day to day. So with the development of fMRI, a lot of people said, well, hey, let's try to become more natural. And this really led to the first big discovery. The retinotopic areas we had known about from non-human primates, but what we had known about was that there's specific parts of the human brain, and it turns out the non-human primate brain as well, that are specialized for processing different kinds of images. So these were studies done by Ina Puce, Jim Haxby, Nancy Canwisher, and others. And what they showed very convincingly is that down in that ventral surface of the brain, the same regions that are color selective, as I showed you a few slides earlier, there's big chunks of brain that respond a lot more to images of faces like this one, and that's in lateral fusiform cortex. And if you go medially, we see there's big chunks of brain that's green color here that responds more strongly to images of houses and places, buildings. And so this was a big surprise, because there'd been inklings of this, but no one had really thought that there'd be such a dramatic dichotomy between these real world categories of images. Because again, if you think view one just breaks down images into oriented bars and lines and luminance edges, then you wouldn't expect that higher areas would have such a dramatic anatomical separation between different kinds of images. So that was obviously earth-shattering for somebody like me who had only ever shown gratings and moving dots and colored wedges. So then you start to ask, well, what about motion? So all of those studies those early studies showed essentially grayscale images of static pictures like this. But what about the dynamic stimuli? Obviously, motion we know that's an important visual cue. What if you have moving visual objects? What happens in the brain then? So we set about making some dynamic visual stimuli. And in the first study, we used whole body movements. So here's an example stimulus. See if we can get this to play. It's always hit or miss. There we go. Okay, so we can name that. What kind of motion is that?
With the jumping jack.
Exactly, yeah. So we had, and it turns out there's lots of different movements you can name. So we had 28 different complex human motions. And the drawback of these naturalistic stimuli are very hard to control. Obviously, it's very hard to make an artificial stimulus that contains all of the properties of a jumping jack. So the general approach taken by most folks is just to create a lot of different natural stimuli and hope that on average, all of the different visual properties kind of average out so that, again, we know retinotopy is important in early visual areas. So you try to have stimuli that cover the whole visual field. Each one stimulus doesn't, but across all stimuli, you try to cover many different visual attributes. So we had these 28 different complex human motions. Obviously, you have another question. Well, yeah,
When you
Johannes.
say that, you mean that you for, let's say, the jumping jack, you've got 100 different examples of jumping jacks at different locations in the visual field? Or are you saying across the entire ensemble that you're hoping?
Yeah, I mean, I think probably we're not closely controlling retinotopy here. So, but other aspects, you do the best you can. That's why we have a gray background in these videos, but it's a big problem. You just can't do it. Basically, you can't control all these low-level cues that you can control so nicely with these
I guess
artificial
before my
stimuli.
question, you're not having multiple different movies as examples of jumping jacks and multiple movies of examples of sitting on a chair.
Well, we do have 5 or 10 movies of each movement type,
Okay.
so there's not just one.
Okay.
But yeah,
Got it.
even
Okay.
within the jumping jacks, there's a limited range.
Okay.
So, but Johannes' question raises a great point. Well, what do you use as a control for this? Obviously, the brain is going to activate when you show these videos. What do you compare that with? Well, what we compared it with was handheld manipulable objects. I won't go into this, it's a whole lecture in itself, but we know that over millions of years of human evolution, we've developed a penchant and a specialization for we have opposable thumbs, so we can use tools. And in fact, one of the favorite studies is to go out and find animals that use tools too, and say that, humans aren't the only ones that use tools. But it's pretty safe to say that we use tools orders of magnitude more than any other animal in the natural kingdom. And there's neuropsychological data, including this PET paper from Alex Martin, showing that if you show static images of tools, you can see activation in the brain. One of the interesting things about tools is that they're defined by their motion. So I can show you this video of a hammer, but I could take this mouse, if I was in a bad mood, and start hammering it. And you would say, what am I doing with that mouse? Well, he's hammering with the mouse, because what defines hammering is not actually the shape of the object. It's that motion that you make. Khrushchev famously hammered the podium with the shoe at the United Nations. And no one would normally use a shoe for hammer, but that motion is what defines hammering. Even if I had a very weird looking hammer, if it was making that motion, you'd say, oh, it's hammering. And even though you can't see a nail or any other implement. So this is a way to show another kind of complex object that's moving in a characteristic way as a control for these biological motion. Control is a strong word, but we're also interested in what's active for these stimuli. And we have 28 complex tool motions. That was lots of fun to make these videos. And when you put folks in the scanner and show them these different kinds of videos and you have them do a matched behavioral task so that they're attending to them, which is important for all of these studies, what you see is just like with faces and houses, you see a very broad network of areas. The image is quite clear on my screen, but on this monitor, it's a little blurry, so apologize for that. But you can get the gist of it, which is that here on the ventral surface of temporal lobe, just like before, you see these lateral fusiform areas, which were very responsive to faces, are also very responsive to whole body movements. There's a middle area here, not quite as medial as the place selective areas that are responding to tools. And then in lateral and middle temporal gyrus, there's some more, the blue colors here correspond to greater activity for tools. And then up here in middle temporal gyrus, superior temporal sulcus, lots of activity for body movements. And then in parietal lobe, lots of activity for tools. So a very complicated network of areas, again, large scale areas that prefer these different kinds of visual cues. And that's, again, unexpected from say the work in non-human primates, where the focus was on very small scale organization. So we tried some other experiments. I won't go into all the details today, but you can make these so-called point light displays where you remove all of the form and color information and you just try to leave that motion information. So there you can see a hammer hammering, and you can have, in some cases, the exact same number of dots between a tool video and a human video, but we're remarkably good at just using that motion information to say, oh, that's a jumping jack. Even though I've taken away the image of the body, the fact that there's a face, there's no dot on the head here. So your brain is very good at extracting that information from just the motion of these point lights. And so the general idea here is that we have this dorsal pathway, which corresponds to this blue stuff up here, which is very important for manipulating things, for spatial orientation. Sometimes it's called the where pathway. We have this ventral pathway, which is often thought of as object form and identity. But the idea is there's also this lateral stuff here, which isn't well captured by either the dorsal or ventral pathway. And it's not clear exactly how to describe it. In a review, Dave Pitcher and Leslie Ingerleiter said it was a social perception pathway. I think it's more multisensory than anything else, because I think that better captures what's unique about these areas. But clearly it's definitely not just about space like the dorsal pathway or about object identity, because if you compare moving and still objects, as we did in some of those studies, I won't show the data, but the ventral pathway doesn't care that much if the object is moving or not. But these lateral areas, most importantly, area MT, which is back here in this little blob here, cares a lot if an object is moving. It responds much more if a real object or a gradient or whatever is moving compared to still. So these lateral areas really care about dynamic moving stimuli. So just to review about temporal lobe, this is just cut off the temporal lobe here from the rest of the brain. You can see there's middle temporal gyrus. Superior temporal sulcus is shown here. That's one of the second biggest sulcus in the brain, actually, after the lateral sulcus. So a huge amount of cortex buried in the sulcus. And then if we look on the top side of temporal lobe, this is Heschel's gyrus here, and that's actually where A1 is, the core part of auditory cortex. So it's very provocative to say, well, and again, in this review, they call this a visual pathway, but boy, this is all auditory cortex right here. So why would this be just a visual pathway? So what's happening with the auditory modality in this region? So that led us to do some studies where we, in addition to just showing the video of the hammer, we could play the sound of the hammer and ask people to identify objects from sounds. And people aren't as good as identifying from sounds as they are from videos, but they can do quite well depending on the object, but certainly 80%. And then we can say, well, what's happening in the brain when you identify objects from vision, the visual modality, or the auditory modality? And what you can see, if we look in auditory cortex, here the visual objects or pictures of visual objects. Here we just have control scrambled objects. Auditory cortex actually is deactivated. So it's shut off. It goes below baseline relative to looking at a blank screen when you see a visual object. Whereas when I play an auditory, or a recording of an auditory object or a controlled scrambled auditory, we see a robust response in auditory cortex. Visual cortex, we see the mirror response, the mirror image response, big response to visual objects, smaller response to scrambled visual objects, and actually a deactivation to these auditory recordings. In contrast, when we look in between, so auditory cortex here in blue, visual cortex here in orange, when we look between auditory and visual cortex in the superior temporal gyrus and sulcus, we see a response that looks like this. There's a response to visual objects, a response to auditory objects, but very little response to these scrambled controls. So this is a part of the brain that really cares about both modalities, but prefers real objects to scrambled. And that was always, it's always been a consistent finding. You can take the best simple stimulus you can think of, like a grating that knocks you out when you look at it, makes you dizzy, but these higher level visual areas just don't care that much about it. They're really specialized for real world objects that have ethological relevance.
This is still the response to single modality presentation.
Yeah, so that's great. Entree to the next slide, which is, well, what if we present the bang of the hammer, the video of the hammer, and then present them together? And you can see that in this STS region, there's actually an enhanced response. Auditory's here, visuals here, and then when I present them together, you see an enhanced response. And that's also a sort of signature of these multisensory areas. When they're integrating, you see a bigger response in either modality alone, whereas in auditory cortex, we don't see that. It's basically the exact same response to auditory only. So anatomically, it's a fascinating question where these regions are. As you can see, if I map them on a single subject, this red line here is where the action preferring, the body movement preferring regions are. The blue is where the tool movement preferring regions are. You can see for the for the tool movement preferring regions, the visual tool regions, there's actually a pretty healthy auditory response there, but not nearly as big in these visual tool response, in these visual region, the body movement regions. The response is fairly anterior compared to the visually selective body movement regions. And that makes some sense because you can recognize tools from their sounds, okay, but you can't really recognize body movements from their sounds. So if you hear someone doing a jumping jack, it doesn't sound that different from someone doing a push-up. I mean, besides the grunts and the sweating, but so there's not very much auditory information there. So what is there auditory information in, or what kinds of visual stimuli come along with a lot of auditory information? And one example, of course, is speech and facial movements. So if you close your eyes and you hear me yawn, you have a pretty good idea what my face is doing. Or if you hear me say a word, you have a pretty good idea of what my face might be doing. So then that raises the question, well, what about these yellow areas? They don't respond a lot to recordings of tools like a hammer hammering, but we know from other work, especially the work of Pascal Belin colleagues and Emiliano Formisano, that there's voice selective areas in this part of superior temporal gyrus. So just like in lateral fusiform, there's brain regions that respond preferentially to images of faces. In these superior temporal gyrus areas, there's regions that respond preferentially to recordings of voices compared to any other stimulus you can think of. So they do respond a little bit to cats meowing and dogs barking, but not very much to hammers hammering, phones ringing, and things like that. So then an obvious question is, what's the relationship between these voice selective areas and face selective areas defined visually? So to answer that question, we did an fMRI experiment where we compared the response to visual stimuli, which were faces moving, either mouths moving or eyes moving, and auditory recordings of stories, vocal sounds, and non-vocal sounds. And our prediction is that if this is really a multisensory area that's putting together information about faces and voices, those parts of the STS that respond to mouth movements should also respond to voices, because that's where the information overlap is. If you hear me say something or you hear me yawn, that gives you information about what my mouth is doing. It doesn't give you information about what my eyes are doing. And in fact, when my eyes can move a lot and there's no sound associated with that. So mouth movements conversely come associated with sounds, whereas eye movements do not. So the prediction is that there should be this correspondence between auditory responsivity and visual responsivity, but only for mouth movements. So we had videos like this. Let's see if we can play one. So here's Lynn and Muge, two students involved in the study. So here you can see them making in those two clips, they made mouth movements. And then in these following clips, hopefully these will show up. There you can see there's Muge's making an eye movement, Lynn's making an eye movement. So it's the same faces, but the type of facial movement is different, either mouth movement or an eye movement. Johannes is thinking, what about retinotopy? Eyes are in the upper visual field, mouth is in the lower visual field. We also did a control experiment where we masked out just the eyes or just the mouth and showed them both right at the center of gaze to make sure it wasn't just an upper visual field, lower visual field kind of thing. And then the auditory speech stimulus, I won't play it for you, but these are just short stories accompanied by a blank screen. And what you can, oh, maybe I should, well, I don't think you need to hear Aesop's fables, although they are educational. So, so if we look at what these maps look like in individual subjects, I'm just fading out all of the stuff outside the superior temporal gyrus. But you can see in this part of the brain, you have more anterior areas that respond strongly to mouth movements. So those videos of mouth moving, and more posterior areas responding more strongly to videos of facial eye movements. And now if you look in these two different parts of posterior STG, you see a very different response to auditory stories. So that more anterior mouth selective region responds very strongly to auditory stories, but the eye ROI responds basically not at all, with a mean of about 0. So that shows there's an overlap between the preference for visual faces, and especially mouth movements, and auditory stories. Okay, so
I got a quick question.
yeah.
So you mentioned retinopathy. I want to come back to it. It's kind of boring, but I want to ask: are they fixating patients, the subjects? Visually?
Short answer, no. you can, in the control experiment I told you about, we had a very small aperture where the mouth and the eyes were in. So then their eyes were probably moving a lot less because it was a 1 degree by 2 degree or so window. But when you show people faces, they make very characteristic eye movements, as Yarbus and many others have
You don't
studied.
necessarily control it. Do you think that it matters in terms of these responses, if they're not looking at the, for some reason, they're not interested in stimulus, they're looking for areas. So they determine which condition they're in, so they're not unaware of the condition. But they're not actually foveating or using that high acuity that's presumably important to their innocent things.
Yeah, I mean, there's a bunch of different levels. So one question is obviously, is it the case that there's more activity? Is this dissociation because there's a difference in eye movements? So that when someone moves their mouth, I look in the upper visual field, and when I look at their eyes, that's the lower visual field. So this is just because there's a visual field map, and this is a map of, this is one big retinotopic map, and all I'm showing here is retinotopic. We don't think that's true, for instance, because we did that control experiment I told you about. I can do retinotopic mapping all day, and I won't see a nice lower visual field boundary here. Are the eye movements different in the two conditions? They are different.
I'm mainly thinking about the role of photoacuity in any of this.
Yeah, so people will naturally foveate facial movements, but it's a little chaotic. People these faces are big enough that you don't necessarily need to foveate to see what's going on. So people can, say, and so we have done experiments where you force people to foveate the nose, say, and you can still get mouth information and eye information even if you're foveating the nose. And you still see most of this stuff doesn't change.
Okay, we'll come back to maybe later.
Yeah, I mean, it's a fascinating question. It gets back to this control naturalistic thing, because again, we don't naturally just foveate the nose. We naturally are doing all sorts of visual sampling. So it's a question of how tightly you want to control behavior at the cost of making it less natural. The stimuli also look kind of weird because you end up with a fixation cross right on the nose, but definitely something that we think about a lot. So, one of the classic criticisms of fMRI is like, well, this is great, you have all these blobs, but who cares, right? Like, does this actually do anything? And that's a very important criticism, because we don't know with fMRI, which is a purely observational measure, whether all these stories that I'm telling you about mouth and eye regions, I mean, it's great that there's that overlap and suggestive and so on, but does it actually do what I'm telling you it does? And in humans, we have a limited repertoire of tools that we can use. We've done work with patients, trying to find patients that have lesions of these areas. I won't show you that work today. That's a classic tool. Another tool that we can use is transcranial magnetic stimulation. I will tell you about that today. But before you can do any kind of causal kind of study, you really need to have a sensitive behavioral measure. Because when I say, it's not realistic to expect that if I show you an image of a face, whatever I do to you, you're probably still going to be able to recognize that it's a face, right? So you have to come up with a more sensitive measure of perception than just, is there a face on screen or not? Because you really have to do a lot to somebody to make them unable to see a face. So that's why we spent probably the majority of our research time, or I've spent the majority of our research time really trying to dig into this behavioral assay so that we can answer this question, is this blob actually important? What is it doing? So fortunately, that you don't have to start from scratch, because long before there was fMRI, people were studying auditory perception, speech perception, visual perception. So you can go and say, well, it's been known for more than 70 years that visual speech can serve as a natural hearing aid. So that if you see the face of someone talking to you, you do better at understanding them than if you just hear their voice. So I'll give you a demonstration of that. Now, if I play this, and of course, to enhance the demonstration, we can add noise to the speech, because if there's no noise, it's pretty easy to understand speech. But if you're in a crowded room or you're at SFN, you're on a crummy cell phone connection, it can be quite hard to understand someone. So I'm going to tell you that this is one of these 3 syllables, ba, dar, ga, but I've added some noise to it. So people, if I play that, are pretty much at chance on whether it's ba, dar, ga. Now I can present the visual modality alone. So this is Taylor, a med student, recording that syllable. Maybe hard for the folks on the other side of the room. Now you can see her mouth is open when she's pronouncing the syllable. You can't pronounce ba with a open mouth. So then you know it's da or ga, but you're still, sort of 50-50 between da or ga. But now if I combine the auditory and the visual, let's go do that over here. What do you guys think? Ga, Beauchamp says ga. Ga, anyone? Ga, da, who thinks da? All right, okay, we've got some das and gas. So it's ga, and that's about, again, about 75% accuracy. People will get ga for that, compared to 33% over here, which is auditory only. So you can get quite a big increase in accuracy when you can see the face of the talker. That's also true, not just for syllables, it's also true for entire sentences. In a nice twist of fate, I think Seth is on the call on Zoom. So Seth is a postdoc in Brett Foster's lab, who many, many years ago, when he was an undergrad, recorded some stimuli for Bharat Chandrasekharan. So if you see Seth, you can give him a hard time. So this is Seth saying a sentence. Very hard to understand. When you see Seth talking, it's the team played a difficult game. So just like with the syllables, people will go from pretty much, basement performance to getting 2/3 of the words, right? Something like that, maybe half of the words.
You need to foveate mouth, yeah, right?
Well, the one question, when you're talking to someone, typically you're at about arm's length. So then the face is maybe 10 by 5 degrees, something like that. And then you could be looking at their eyes and still get mouth information because but here people will naturally, when you play them noisy speech, they always fixate the mouth.
Get it. But it's important, what I'm proposing.
Yeah.
Way, could there be a perceptual deficit?
Yeah, so in a case like that, again, it's quite sensitive, right? Because your acuity falls off pretty sharply. So if I have you fixate the nose, you're still getting enough visual information from the mouth that you get that visual benefit. If I put the fixation point up here in the top of the head, then you're getting out into the paraphobia and you're going to start to lose that. But it is actually fairly coarse. You can also do things like spatially filter the face. So if I low pass filter the face, you can still get some visual information out of it and get a visual benefit. So it's not just super high spatial frequency stuff. There's a lot in just the timing of the movement, for instance, which still comes through in the low spatial frequency. And you can get that fine from magna cellular pathways. So that's why I mean a little hedgy here, because I don't think it's just foveal, parvo, high spatial acuity.
I'm not surprised.
Yeah.
You explain something that you developed, what it different or is it actually
No, it's the exact same auditory clip in both cases.
in the previous?
Oh, yeah, so in all three, here, this auditory is exactly the same as that auditory. All that I've done is add visual information.
Right, but I didn't understand why the right position is better than the pure visual position. The auditory actually have some signal to it or else?
Yeah, it's a recording of ga with noise added.
That's what I meant.
Yeah, I mean, I've added lots of noise, so it does just sound like noise, but it's ga with 10 dBs. I mean, it's ga minus 6 dBs of noise.
Okay.
And then when I add the vision, that's enough to lift the signal out of there. I apologize, that AV is truncated there, so that should say AV, but this is, again, this has been known since 1950s at least, so this isn't a new discovery, but I think it's worth repeating that this is a very reliable observation that we use every day. So whenever you go into a loud restaurant, your brain is forcing you to fixate on people's mouths so you can understand what they're saying. And if the restaurant turns on the lights, then you're really in trouble. So this is
Because you're staying on everyone's mouths.
sorry,
Why are you in trouble when they turn on the lights?
turn off the lights.
Off.
They dim the lights on you or in a nightclub or whatever. I mean, since we're all going to nightclubs all the time. Or it's dark out.
Got it.
They do that at SFN, to get all the scientists to leave their posters, right? They start flicking the lights off at the end of the poster session. Okay, so this is great, very reliable, replicated hundreds of times. It's not great for psychophysics experiments or neuroscience experiments because you need lots of trials to make these graphs. So what we want to do is run some kind of neural interference experiment like TMS and see if we can eliminate this behavioral benefit of seeing the face of the talker. But people don't like these kinds of experiments. Like Russell said, they're like, there's nothing in there. What are you trying to do? You know, trying to torture me, make me guess at words that I can't hear. So very common reaction. So it's not great for patients, not great for kids. So that's the segue to the McGurk effect, which Johannes mentioned in the introduction. And the McGurk effect is beautiful because it's so simple. So this was first described in a Nature paper in 1976. Harry unfortunately passed away in 1998, but John McDonald is still alive and kicking. This is him at a conference in 2016 with me. I'll show a video of Harry McGurk doing the McGurk effect. He's an irascible fellow, so don't be alarmed by the emotional tone here. Yeah. And this is why the grad students are unionizing. Oh boy, I'd be a unit too. John was his research assistant, so he tells good stories about Harry being irascible. So in any case, we don't have any videos of the original McGurk effect. This was not what they used in that Nature paper. Those were an actor that they got to record it because Harry divorced his wife and moved to Australia, and his ex-wife threw out all the stimuli. So no one knows what the actual original McGurk stimuli look like, sadly. So everyone makes their own stimuli. This is a fellow, Arndt Mosso, who's a professor at the University of Oslo, and he's made some stimuli and posted them on YouTube that are about the most watched videos, the most watched McGurk videos. They're about 1 million views. YouTube can't be wrong, so those are the ones I'm going to show you. So again, now the nice thing is we don't have to show any noise. I'm going to play the auditory track. Ba, 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 ba. And you should all hear ba, 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 ba. There's no question that there's nothing there. Now I'm going to play the video of what Arndt Mosso is saying. And again, it's a little pixelated here and hard for you to see and everything, but visually he's enunciating the syllables ga, 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 ga. So the McGurk effect is that when I combine that auditory ba ba with the visual ga ga, people often perceive da da, which is what I'm going to show you in the next slide. Ba 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 ba. And it's quite a powerful illusion. People say, oh, you're lying. But if you just shut your eyes and hear, you can close your eyes and I'll play that same clip. Ba 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 ba. And it will sound like ba ba. So it doesn't go away. So then you have this very powerful illusion. And the nice thing is I can assay that in a few seconds, right? So instead of 100 trials of noisy speech, I just play 2 seconds of Arndt Mosso here and say, do you hear ba or da? And
you're playing ba, to be clear.
The auditory is ba,
Definitely.
the visual is ga, and then the fusion percept is da. So if you're blind or if you have your eyes closed, you hear ba. If you're integrating auditory and visual information, then you perceive da, typically.
My line of questioning is essentially around the role of orienting attention and eye movements, and is conflated with foveation, which you've already talked about, and Brendan Tuffy. But more importantly, it's related to this idea of engagement. If they're not staring at the stimulus and they look away, do they lose the perceptual integration?
Yeah, I mean, it's an interesting question. People have tried to do that experiment. Faces are quite attention grabbing. So it's hard. People will naturally tend, and it only takes a short fixation or attention break to look at the mouth to give you that enough information to say, oh yeah, that's a visual ga, I'm going to go with da. So attending away from this face will definitely reduce the McGurk effect. It doesn't eliminate it entirely. It's impossible to say, it's difficult to say, is that some information slipping through, getting to the STS through the information filter, or is it some micro attention break?
You might be able to say something that's relevant to your concerns here, though. I mean, this is something I use in my undergraduate class. And so what I have them do to modulate what the percept is to focus on the mouth and then look progressively further away. And you can actually just change the perceived syllable as a function of how far away you're fixating from the mouth. And you can also modulate in the other direction towards the visual or towards the visually indicated person by turning down the volume of sound. So you can still hear the sound, but then it'll start sounding more like a ga than a ba, just because the auditory information is less reliable. So you can push what the perceived syllable is in both directions by changing the reliability of the signals, each of the two signals.
You just give me great segues, yeah. So I was just gonna talk about Bayesian integration, and that's exactly right. As your brain is just trying to decide, I have this incongruent information from the two different modalities, what should I do? The idea is that you settle on DA, your brain settles on DA because DA is sound audit. If we looked at the spectrogram, it's similar to BA. So it has some auditory consistency with BA, but it has that open mouth shape. So you can pronounce DA with an open mouth, just like you pronounce GA. Whereas you can never pronounce da or ga with a closed mouth. So your brain says, okay, the da is creates the least Q conflict between the two modalities. So that's we model it more quantitatively than that, but that's the hand wavy explanation of why you end up with that fusion percept.
Does it matter how synchronous they are?
Oh, of course, yeah. So we've done experiments on that. The temporal integration window for speech is quite broad. It's a couple hundred milliseconds, but it's asymmetric. So as long as the vision is coming before, and I'll talk about this in a little bit, as long as the vision is coming before the auditory, you're okay. But as soon as the visual comes after the auditory, then you lose it because of this causal inference problem. Okay, so now that we have this nice simple assay of multisensory integration, we can test this hypothesis that if this part of the posterior STS is where this multisensory integration is happening, it's taking the BA auditory information that's coming in from auditory cortex, the GA visual information that's coming in from this motion stream here, and putting them together to produce this DA percept. Then if I interfere with activity in the STS, then I should be able to reduce or eliminate the McGurk effect. So that's the study that we did some time ago now. So first we scanned subjects to find out there's lots of anatomical variability in this part of the brain. So we identified this multisensory region in each subject. Then we used image frameless stereotaxy to manipulate the TMS coil so it would be right over that region. And then we applied single pulse TMS as subjects watched the McGurk stimulus. And what you can see is that when there's no TMS, and I will emphasize that these are subjects we selected to get the McGurk effect, because some folks don't get the McGurk effect, and obviously then there's nothing to eliminate because they don't get it to begin with. So we selected folks who got the McGurk effect strongly. Then when we delivered TMS, you can see that how often they got the McGurk effect dropped precipitously by about half. And then when we did TMS at a control site, TMS makes a loud click, it makes your scalp tingle, so that's a concern. When we did TMS at the vertex, we didn't see any effect, so it can't be attributed to some of the other side effects of TMS. That was for the ba, ga, das syllable. We also tried the other McGurk syllable, which is auditory pa, visual ka, often produces the percept of ta. Same thing. Now, if we change the timing of the TMS pulse, this gets back a little bit to the asymmetry I was mentioning before. The TMS is only effective in a window of about 200 or 300 milliseconds, slightly before and and a couple hundred milliseconds after auditory onset. So that's when this integration is occurring according to this measure.
So the visual needs to come in first,
Yes.
then the auditory is necessary because if you lose either one, you lose the effect.
Mm-hmm.
And then if you zap this integration site, putative integration site, at any point after, essentially when the auditory comes in.
Yeah, so from 100 milliseconds before auditory to 100 milliseconds after. Or maybe that's 50. Yeah, I think we did 100, 0, 50, 100. So 100 before to 100 after.
You're disrupted.
Yeah.
But if the vision had to come first and it had come at the same time as the auditory onset,
Yeah, the McGurk effect would be very weak then.
then you
And
wouldn't get the effect. It'd be like then it would all be blood.
yeah, then it would
There'd
all
be
be
no
low.
effect of this then.
Yeah, so if people are down here, then you can't measure anything. So this was, I thought this was a great result. My collaborator, Tony Rowe, who is the TMS, who started me on TMS, left shortly after. So I've never done another TMS experiment after this one. I was very glad to see last week that Brett pointed me out to this, to David Brang, who's a professor at University of Michigan. So he recently replicated this finding using of a dissociation in the McGurk effect using TMS. So that's a relief. It's always a relief when your studies replicate. Okay, so in the interest of time, again, I'm going to skip some of the modeling studies and the behavioral studies, and I'll just close talking about some intracranial EEG experiments. And so these are experiments that we're doing right now over in the pavilion on the 9th floor of the very beautiful hospital here. And this was work that was done in collaboration with Dan Yosher. Here's a picture many years ago when I was still at University of Texas and Dan was at Baylor. And these are patients who are in the epilepsy monitoring unit with electrodes implanted in their brain for their clinical treatment, but they're just sitting there with not much to do. So if they're in a good mood, fingers crossed, they can agree to do some of our experiments. And recently there's been a transition in clinical practice from these ECOG electrodes that sit on the surface of the brain to stereo EEG electrodes that penetrate into the brain. And here you can see each of these white hyphen looking things here as one contact on these penetrating electrodes. That's been a bit of a setback to the research effort, because in the old days we would get lots of electrodes that covered this part of the brain. Now we're lucky if we get a handful. So, but we have to keep charging ahead. And the typical response that we measure from these IEG experiments is this so-called BHA, broadband high frequency activity response, which is not phase locked to the onset of the stimulus, but continues on as long as the sensory stimulus is present, and it covers a pretty broad spectrum of the frequencies band. And we think it reflects the summed firing of lots of different neurons. And our model of the neural computations that are happening in posterior STS is that we have small populations of neurons that are responsive to specific phonemes. So this is work from Nima Mesgrani, Eddie Chang, and many others. And the idea is their selectivity profile has some roll off. So they're most responsive, say this pool of neurons might be most responsive to the ba sound, but it would still respond a little bit to other sounds. So that if we presented a ba sound, the ba neurons would fire the most, but other neurons would still fire a little bit. And then you have some downstream area that does a winner take all selection mechanism and says, okay, the ba neurons are firing the most, that must be a ba phoneme that I'm hearing. So the idea for multisensory integration is that visual mouth shape that you get when you're foveating the mouth of the speaker, or at least it's in somewhere near your foveal vision, is informative about the auditory sound. And the way that the physics of speech work is you make that mouth shape before you start vocalizing. So I need to make a ba mouth shape or a da mouth shape before I start vibrating my vocal cords. So when I see that mouth shape, that tells me, as an observer, that tells me that the phoneme that I'm going to hear a short while later can't be any of these red phonemes. It has to be one of these green phonemes. So it's not a one-to-one mapping. There's fewer visual mouth shapes or visemes than there are phonemes, but it still is quite informative because there's many phonemes that I can rule out. So that's the beauty of multisensory integration. I can use this Bayesian inference process to say, okay, given this prior of the mouth shape, I can downweight the probability of all of these red phonemes. So you can imagine if I have a noisy auditory stimulus where you're not sure what that phoneme is, all of the neuronal pools are going to have a pretty weak response. And your downstream area is not going to be able to figure out what to go with, and you'll just say, I don't know. When I have that visual mouth information like I'm showing here, then you say, well, that's a ga mouth shape, and maybe it is compatible with, and that means a lot of these other sounds like ma are incompatible because you can't make a ma sound with the ga mouth shape. And so that if you enhance the activity of these green neurons and inhibit the activity of these red neurons, then when I do the auditory and visual together, I'm going to get a response that looks like this. And then the downstream area can say, oh, that must be a ga sound, because there's a big enough difference between the ga neuron responses and the other neuronal response pools. And so I can make this perceptual decision and say, okay, here I'm below the decision threshold, don't know what it is. Here I'm above the decision threshold, and I can say it's a ga. So that's sort of our working model of how multisensory integration works in this part of the brain. It's harder than you might think to test this model because we don't have neuropixels, at least not yet, that let us go in and measure individual neurons. We're still stuck measuring the summed activity of many neurons. And I won't go through how this would apply to visual, to incongruent stimuli like the McGurk stimuli. But the basic idea here is that we can measure the sum response of many pools of many neurons, because we can't measure the individual neurons. And the idea is that if I just have auditory syllables, a lot of these pools are going to fire to some extent. So I'll get a pretty big summed response. If I have visual information, I'm going to suppress the response of a lot of those neurons because they're incompatible with the visual information. So when I take the summed response of all of these pools of neurons, then I'm going to get a weaker response than when I just have auditory information alone. So that's the tool that we're using to measure indirectly what's happening in these phoneme selective populations of neurons. And I wouldn't be telling you this nice story if we didn't have experimental data to support it. And the experimental data is first that when we go into this part of the brain with IEEG, we actually see this suppression that was predicted by this model, where the audiovisual response is actually weaker than the auditory response. That could be non-selective, could be due to a lot of different things, but we can show that that's not the case by testing different phonemes and words that have a head start or not. So it's not always the case that the mouth opens before the sound comes out. With a sound like there's actually no mouth movement. So you start hearing that sound without seeing any visual mouth movement. So this model of suppression predicts that for a phoneme like we shouldn't see very much suppression, because you don't get any visual information that tells you, hey, it's not going to be this phoneme or that phoneme. And so in this interval between the visual mouth shape and the auditory information, you can enhance compatible and suppress incompatible phonemes if that information is available. And so that's the experiment that we did here, where perceptually we show that if you have words with a sound where there's not much visual information, you get less benefit from seeing the face than words like drive and last, where there's lots of visual information. And correspondingly, when we look in the brain, we see a greater neural suppression when you have these words like drive and last that are visually informative, compared with words like known and meant that are not visually informative. So that provides some evidence, at least, that this suppressive model could be explaining the multisensory integration interactions in posterior STG. Obviously, lots more work needs to be done.
And it's based on suppression of the distractors or the incorrect.
Yeah, the incompatible phonemes.
Sorry, I must maybe miss something, but on what basis are you saying there's no good visual information about known or meant?
Well, if you just
I guess there's two questions. Yeah, that's the general question. And then is there a quantitative assessment of that, or is it how are you grouping with that?
Yeah, so the idea here is this green, and sorry, I'm rushing a little bit because as usual, when you have a new talk, it takes longer than you think. The green curve here shows that for a duh sound, the mouth opens quite some time before this purple curve starts to rise, which is the auditory amplitude. Sure. Whereas if I made the same curve for the word meant, then the green and the purple curve would start to rise at, there's very little green, the green curve would be quite low, and it wouldn't change until the purple curve had already started to rise. So you don't have that head start of the visual information telling you about
what in that sense that you mean. So this asynchrony between the onsets.
Exactly. We think that's what's helping providing that information, because the auditory processing happens pretty quickly. So if 200 milliseconds later, you're like, that was probably a mouth shape. I mean, that could have been a mouth sound. Maybe that's too late to help you. I mean, there is some benefit. So it's not the case that there's no benefit for perceptually, but it's certainly a lot less than for these words where you have the head start. So I'll just finish with a few outstanding questions. Obviously, we still don't know what the detailed neural computations are. It's still a puzzle to me why in fMRI we typically see enhancement when we have multisensory. So in fMRI, this orange curve is less than the blue curve, but in IEG, this orange curve is greater than the blue curve. So that's kind of a mystery. Still trying to figure that out. We don't know what the computations are, the inhibition and excitatory computations. You don't really need both. It could just be excitation, or it could just be inhibition of the incompatible phonemes. So the detailed computations, and at the large scale, we know there's lots of other brain areas besides, after this sensory step of saying, okay, is the person smiling at me or frowning at me? Are they saying beer or deer? You have to take that information and pass it to the rest of the brain to say, okay, they're frowning at me, maybe I'm in trouble, or they're smiling at me, maybe I should smile back. So there's lots of other interesting social computations that have to happen with this information you're getting from the SDS And I haven't talked about that today. There's lots of other interesting things going on, turn taking and so on. So it's a very, and I was fascinated to see Michael Platt's finding that monkeys on his island in Puerto Rico keep track of how often they groom each other over months. So it's always fair. So if you groom me for 5 minutes today, I remember that 2 weeks later and that it's my turn to groom you. So obviously we're very, always keeping track of stuff, and that's obviously probably not happened in the STS. So non-human primates could be a good model for understanding some of these social interactions. So I'll finish again by thanking all the folks who did the work, and the funding agencies, and reprints for all the stuff is on the website. Thanks very much. Yeah, Alan.
At the end, you said, you see this kind of opposite effect if you look at IEG. So you were recording with fMRI at the same time? Or?
No, this was all.
I mean, the resolution is not, you can't do that. That's true.
Yeah, we did not do these experiments at the same time. So I'm not saying fMRI and IEG are different. I'm not making a big claim about anything. I'm just saying it's one of those things that irritates me that I don't know the answer to. So
No, but you said you see the opposite polarity. But I'm assuming you're not looking at exactly the same experimental settings.
exactly, it's a different setting, it's a different modality, it's a different. We've not done the exact same experiment in the same subject in the exact same brain region, which you'd need to do to say that it's really, to get at that difference.
So you could not decode the syllable on the fMRI signal. Try.
Well, Formiziano has a science paper where he decoded the syllable,
Okay.
so you can do that.
It would be, in principle, you could run the same.
Yeah,
Okay.
yes. I think that would be good to do. Yeah, question.
Yeah, very interesting. If you just look at the areas dedicated to either the visual or the auditory processing of MRI, probably a lot of variation. Do you find that these variations are positively correlated, or if you have a bigger area devoted to the auditory, there's less of an area devoted to the visual? Do they trade off one to another, or are they both big in some people and small in other people?
Yeah, great question about individual variability. There's a talk at 3:30 today by Randy Buckner on precision neuroscience. So he's going to talk a lot about individual differences, something we think about a lot as well. It turns out the early sensory areas are fairly consistent from subject to subject. Where your V1 is, where my V1 is pretty consistent. There is some variations. Jeff Boyne had a paper showing that if your V1 is huge, you have slightly better visual acuity than a small V1. But we think the real action is in association cortex. So that your STS is probably structured quite differently than mine, depending on our developmental trajectory. And it may not even be so much the size of the area, but the connections that are built there, so that you've had more. For some people don't get the McGurk effect at all. I haven't talked about that today. What's happened in their STS, right? These are healthy English-speaking college students. They don't get the McGurk effect. What's up with that? So it's probably not that they have an STS because we've scanned them, but we haven't been able to find what exactly in the brain is different so that they don't get the McGurk effect. But fascinating question.
And have you looked to see how this interacts with, say, show somebody the letter B? Is there another area that's being pulled in to recognize the shape of the letter B when they're reading? Yeah,
so there's what's called the word form area, the visual word form area, which I haven't talked about today. And that's the FFA is sort of shown here. It's actually on the ventral surface. The visual word form area is right here, just on that junction between the ventral and lateral surface. So that's where that text information gets into the system. Yeah, so definitely connects there. We don't think that language is new enough. Written language is so new that that's probably not an evolutionary adaptation, but for whatever reason, it consistently ends up there in people. All right, thanks everyone. Appreciate it.